Hello students. So today we're going to proceed a new unit called Biotechnology. So in this unit we have two chapters. So one is Biotechnology, Principle and Process, and other one is Biotechnology and its application. So first we're going to deal with Biotechnology, Principle and Process in brief. What is this biotechnology? Bio, it means life, the technology we were used for the desirable product, the technology we use, the technique we implement in the biological process. Biotechnology is a broad area of biology involving the use of living system and organism to develop or make product. Depending on the tool and application, it often overlaps with related scientific fields. In the late 20th and early 20th centuries, biotechnology has expanded to include new and diverse science. For example, genomic, recombinant gene technique, applied immunology, and development of therapies and diagnostic tests. So let us going to deal this biotechnology principle and process in brief. What do you think of when you hear the word biotechnology? Maybe the things you have seen in the news, such as Dolly, the cloned sheep. So this is a snap of the Dolly, the cloned sheep, genetically modified organism, that is GMO. If that's what you think of, you are absolutely right. These are all examples of biotechnology. But what about beer breeding, crop breeding, and the antibiotic penicillin? So these process and product, some of which have been around for thousands of years, are also example of biotechnology. We will first examine that a definition of biotechnology, seeing how it can encompass many different use of organisms to produce useful product. So then we will take a closer look at DNA technology, that is the technique for manipulating and sequencing the DNA. So the DNA technology is crucial to many modern forms of biotechnology. Application of fermentation in the production of wine and other alcoholic beverage is also a biotechnology technique. So now we proceed with definition of biotechnology. Biotechnology can be defined as the use of microorganism, plant or animal cell or the component to produce product and process useful to human. So it is the use of an organism or the component of an organism or other biological system to make a product or process for a specific use. So this is a very broad definition, and as mentioned, according to the European Federation of Biotechnology, that is EFB, biotechnology is the integration of natural science and organisms, cells, part thereof, and molecular analog for product and service. So the term biotechnology was coined by Karl Ehrke in 1919. In 1919, Hungarian agricultural engineer Karl Ehrke foresaw a time when biology could be used for turning raw materials into useful product. So he coined the term biotechnology to describe that merging of biology and technology. So that is the term called biotechnology. So his vision has now been realized by thousands of companies and research institutions. Modern biotechnology using genetically modified organisms was made possible only when man learned to alter the chemistry of DNA and construct recombinant DNA. So we will discuss brief in this lesson. Principles of biotechnology are based on the concept of following techniques. So these principles are include based on the concept. There are two types. One is genetic engineering and other one is advocate maintenance of sterile condition to support growth of only the desired micro eukaryotic cells. So the two core techniques that give rise to modern biotechnology are genetic engineering. So genetic engineering is defined as the direct manipulation of genome of an organism. It involves the transfer of new gene to improve the function or trait. The most important technique of genetic engineering is gene cloning. So next is the manufacturing of antibiotic, vaccines, drugs with the help of maintaining the sterile condition to support the growth of the desirable microbes. The technique of genetic engineering include the following. Creation of recombinant DNA by combining desirable genes, gene transfer, maintenance of DNA in host and gene cloning. Please make a note. Next we proceed with the basic steps in genetic engineering can be summarized as identification of DNA with desirable genes, the first step. Introduction of the identified DNA into the host. And third will be maintenance of introduced DNA in the host and transfer of the DNA to its progeny. How we use these basic techniques in the construction of first artificial recombinant DNA we will see in detail now. It was achieved by linking a gene encoding antibiotic resistant with a native plasmid, that is an autonomously replicating the circulating extra chromosome DNA of Salmonella. So the Stanley Cohen and Herbert Boyer accomplished this in 1972. They isolated the antibiotic resistant gene by cutting out a piece of DNA from a plasmid. The cutting of DNA at specific location was carried out by the molecular scissor, that is restriction enzymes. So the molecular scissor in biotechnology we will call it as a restriction enzymes. The cut piece of DNA was then linked to the plasmid DNA with the enzyme DNA ligase. This DNA ligase which acts as a molecular glue for the technologist. The plasmid DNA acts as a vector to transfer the piece of DNA attached to it. When this DNA is transformed into E. coli, it could replicate using the new host DNA, that is a polymerase enzyme, and make multiple copies of the particular thing. This ability to multiply the copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. Tools of recombinant DNA technology are as follow, that is restriction enzymes, polymerase enzyme, ligase, vector, competent host organism. Please make a note, students. So we will discuss brief in next class. Okay, so please make a note of these tools. Restriction enzyme, polymerase enzyme, ligase, vectors and competent host organisms. So these are the basic steps involved in the process. Isolating genomic DNA, fragmenting DNA, screening the DNA fragment, insertion of DNA in vector, introducing DNA into the host, culturing the cells and transformation of host cell. So these are the basic steps involved in process of biotechnology. Basic step involved in process, the first one is isolating the genomic DNA from the donor. Second, fragmenting the DNA using molecular scissor. Okay, so what is the fragmenting? The piece of DNA is cut with the help of the molecular scissor. Third step will be the screening the fragment. So screen the fragment for a desired gene. Fourth step will be inserting the fragment with a desired gene in a cloning vector. Introducing the recombinant vector into a competent host cell. So this will be the fifth step. Next step will be culturing these cells to obtain multiple copies or clones of desired DNA fragments. And last step will be transformation of host cell. That is, using these copies to transform the suitable host cell so as to express the desired gene. So this is an introduction of basic steps in the process. Principles of biotechnology as we discussed. So that is are of two types. One is genetic engineering and other one is maintenance of sterile environments. So as we discussed before, technique to alter the chemistry of genetical material, that is the dRNA and RNA, deoxyribonucleic acid and ribonucleic acid, okay, to introduce these into host organisms and thus change the phenotype of the host organism. So these are called genetic engineering. Maintenance of sterile environment. So the sterile environment in chemical engineering is a process to enable growth of only the desired microbe. The eukaryotic cell in the large quantities for the manufacture of biotechnology product like antibiotic, vaccines, enzymes, etc. So these are the two processes involved in biotechnology. So now we see about tools of biotechnology. One is restriction endonuclease enzyme, cloning vector, competent host. So these are the three major playing role in the tools of biotechnology. So we will discuss this tool in next class in brief.